Hi, I'm Catherine Holmes, and I'm going to be your narrator today. I've worked with Cell Science System for approximately 20 years, and I'm a certified nutritionist, and so we'll start the program. The Alcat Test is the go-to test for detecting and addressing food sensitivities. This presentation will cover the science behind Alcat, as well as an overview of the protocol used when addressing individual Alcat results. The Alcat test is a leukocyte activation test. It is designed to detect innate immune cell responses to food extracts, food additives, and chemicals. It is not a food allergy test and does not measure or address the presence of antibodies. Later in this module, you will see how the Alcat differs from both IgE antibody testing, which is indicative of true food allergy, and IgG testing, which is more reflective of an actual tolerance to a food versus an intolerance, sensitivity, or an allergy. The Alcat test uses a validated impedance flow cytometry method to detect white blood cell changes that occur in response to a variety of food extracts and chemicals that can serve as triggers and set off a potentially damaging innate immune response. Sustained reactions from a chronically activated immune system can ultimately damage cells, tissues, and organs. While acute inflammation has a distinct purpose in the body, chronic inflammation can contribute directly to disease. Elimination of identified triggers using the Alcat protocol can help reduce the initial innate immune response and with it reduce the underlying chronic inflammation that can be so damaging and help restore bioenergetic balance and function at a cellular level. The human immune system consists of three complementary layers of protective and defensive mechanisms, an extensive system of mechanical, biological, and chemical barriers that act like a coarse filter, an innate, nonspecific set of immune responses present from birth that act as a firewall, a set of specific adaptive immune responses that mature through life and provide a targeted response. Here you can see that the fundamental differences between allergy and sensitivity lie in the different branches of the immune system and immune pathways. Note that an allergy involves specific immune parameters, antibodies, as part of a targeted response, while sensitivity involves nonspecific cellular responses that serve as a firewall and/or a coarse filter. Let's look at the coarse filter. The first layer of the immune system acts like a coarse filter, capturing, destroying, or deactivating potential pathogens by mechanical and biological means. In addition to the protective surface barrier of the skin, many other systems provide a first line of defense against potentially harmful microorganisms and substances. These include mucus, which can trap and help expel invaders, lytic enzymes in saliva, including lysozyme and phospholipase A2, body fluids such as tears and urine that flush microorganisms and other substances out of the system, hydrochloric acid in the stomach, and antimicrobial peptides produced by the skin and the respiratory tract, such as beta defensive and IgA antibodies in the intestines. Here we will describe the firewall more in depth. The second level, the innate or non-specific immune system, acts like a firewall, providing a rapid response against invading pathogens. It can initiate defensive actions within moments of exposure and continues to combat invaders long after the initial breach. In particular, neutrophils act as first responders, offering an acute, nonspecific defense against encroaching pathogens. Neutrophils release aggressive inflammatory mediators, free radicals, and oxidative enzymes, enabling the body to quickly and effectively combat invading pathogens. Neutrophils are vigilant. They continually patrol the body, and by sensing specific molecular patterns, can differentiate between endogenous or harmless bacteria and those that are exogenous and potentially pathogenic. In circulating blood, 70 to 80% of white cells, leukocytes, are neutrophils. Their overwhelming preponderance clearly indicates their importance. When required, neutrophils can extravasate from blood vessels into the surrounding tissues in response to molecular signals of infection, damage, or danger. Here we will talk more about the targeted defense arm of the immune system. The third level of the immune response, the adaptive immune system, allows the body to recognize unfamiliar pathogens and antigens and respond by producing novel antibodies. The adaptive arm kicks in if the innate system's capabilities are exceeded and more targeted approaches are required. It has the capacity to remember its responses, and this remarkable ability helps protect against reinfection and also enables adaptation to new or modified pathogens. The innate and adaptive immune systems can work together and or independently of one another. Ideally, their well-coordinated interactions enable the body to address a wide array of challenges without attacking harmless substances or self-structures. However, ideal is not always, and at times the immune system attacks otherwise harmless substances. In the early 1900s, the term allergy was used to address altered or adverse reactions, a very broad definition. Today, allergy refers to a pathogenic overreaction to otherwise harmless substances that is mediated by antibodies as part of a specific immune response. These antibodies-related responses of the specific immune system are classified as type 1, 2, 3, or 4, according to the Gell and Coombs system. A type 1 reaction, the type commonly tested for, is associated with the release of IgE antibodies and is considered an immediate hypersensitivity response. During the 20th century, science was strongly focused on discoveries linked to the specific immune system. The long history of studies related to the role of innate immune cells, from the discovery of phagocytes in 1882 by Nobel Prize winner Eli Mechnikov, until recently has been largely ignored. However, current research is providing ongoing new insights that overactivation of the non-specific immune system plays a central role in the development and maintenance of chronic inflammatory diseases. Diseases may not be evident immediately. They may develop over a long period of time before manifestation. Food sensitivities can follow different immune pathways, and symptoms may be delayed. Therefore, it is challenging to identify and accordingly is referred to as hidden or masked allergy or diagnostic chameleon. Since inflammation can affect every system and organ of the body, no single medical specialty looks at this phenomenon comprehensively. The Alcat Test for Food and Chemical Sensitivities is a leukocyte activation test designed to identify direct cell-mediated food sensitivity, cellular pro-inflammatory food sensitivity, and or food sensitivity mediated by the innate immune system. We have seen that food can act as immune modulators with positive or negative health impact. In this sense, food functions as an epigenetic factor influencing gene expression without changing DNA sequence. This impact is one of the strongest factors that will determine whether disease will manifest or not. Thus, the intake and quality of food is one of the most potent lifestyle factors that affect us and our health. Knowing that foods can trigger chronic activation of the innate immune system helps clarify the connection between diet, chronic inflammation, and chronic disease. The knowledge that chronic disease are related to a chronically activated innate immune system is relatively a new concept. Hence, not many available laboratory tests are designed to evaluate the corresponding innate target cells. Some sensitivities are governed by inborn errors of metabolism and may remain fixed for many years or even the entire lifetime. However, most sensitivities are greatly influenced by lifestyle factors. For example, poor nutritional status, stress, emotional or physical. Chronic disease may impact the gastrointestinal and immune system and impact the cell's degree of reactivity. Chronic activation of innate immune system results in persistent inflammation, oxidative stress, and perpetuation of disease symptoms. This sustained activation of immune cells is destructive, promoting effects ranging from microinflammation of intestinal tissue to the type of chronic, widespread inflammation responsible for numerous disorders, including gastrointestinal complaints, metabolic disorders, skin diseases, obesity, respiratory disease, neurological, and musculoskeletal disorders. Because activated leukocytes can be present throughout the body, associated symptoms can occur throughout the body as well. As you can see in this table, a number of systems and associated symptoms may be associated with the food sensitivity reactions. Many symptoms and disorders are quite common and seen frequently in physicians' offices, yet underlying causes are often not identified. Commonly involved body systems include gastrointestinal, skin, neurological, respiratory, metabolic, endocrine, musculoskeletal, immune, periodontal. The Alcat test does not look for one single chemical, antibody, or response factor, but instead tries to replicate as closely as possible how the whole leukocyte population responds. With this comprehensive approach, Alcat is able to detect the all-over effect of a sensitivity reaction on various complex immune mechanisms and blood cells. The direct, nonspecific cellular reaction is the immediate first defense and a key feature of the innate immune system. In this reaction, cells initiate and maintain pro-inflammatory reactions and cascades. The innate immune response is not protein dependent in the way that the specific adaptive immune system is. The specific immune system depends on antibody formation and is mainly directed against protein agents. On the other hand, nonspecific cellular defense reacts to a broad trigger spectrum, including pathogens and dangerous molecules. The Alcat test is able to address the multiple factors associated with food, which may not be detected by food allergy testing or antibody measurement. These factors include non-proteinaceous molecules, such as fats, carbohydrates, phytochemicals, xenobiotics, additives, and other processing ingredients that can contribute to adverse food reactions. Here you see what happens when a neutrophil, the most abundant type of leukocyte, is activated by a trigger that may be endogenous or exogenous. The trigger sets off a cascade of reactions, including conversion of oxygen into highly reactive forms of the enzyme NADPH oxidase. In response, the neutrophil will change size, shape, and structure, eventually bursting open. Cell contents are released, including mitochondrial and nuclear DNA fragments, reactive oxygen species, pro-inflammatory cytokines, lytic enzymes, interleukins, tumor necrosis factor, chemokines, and other destructive molecules. This cellular degranulation and associated inflammation have been found to correlate with multiple symptoms and diseases. When single or multiple biomarkers are elevated, the Alcat protocol may help control and eliminate immune triggers. As you can see in this table, biomarkers can reflect a number of immune system activities, including localized microinflammation or systemic inflammation, activation of the innate immune system, cellular bioenergetic imbalance. In humans, 50 to 70% of circulating leukocytes are neutrophils, and thus the predominant immune cell population in human blood. Neutrophils form the front line of defense in the innate immune response to invading microorganisms and microbial structures called pathogen-associated molecular patterns, PAMPs, as well as to foreign particles. Analogous to PAMPs are endogenous molecules called damage-associated molecular patterns, DAMPs, that also trigger an innate immune response. Neutrophils use a number of strategies to respond to PAMPs, foreign particles, and DAMPs. Such strategies include phagocytosis, degranulation, the inflammasome, and the recently discovered formation of extracellular traps. During phagocytosis, internalized pathogens, particles, or DAMPs are translocated to phagosomes where antimicrobial factors derived from granules and reactive oxygen species create a killing environment. Degranulation is similar to phagocytosis. However, during phagocytosis, rather than being engulfed, the pathogens, particles, or DAMPs are killed extracellularly by the same antimicrobial factors that are in part released outside the cell. The neutrophil extracellular trap, nets, can be released by neutrophils in a process called netosis. Nets are a special kind of trap formed by condensed chromatin fibers along with antimicrobial factors delivered by granules. Neutrophils play an especially important role in the body's defense against perceived invaders. Cell fragments and other debris must be continuously removed from the body. This is accomplished by the process of phagocytosis, which literally means cell-eating process. Neutrophils comprise the largest population of phagocytic cells of the innate immune system, representing the majority of granular white blood cells in the blood. The other granulocytes, monocytes, eosinophils, and basophils occur in much smaller numbers. Neutrophils are able to detect potential pathogens because they contain numerous pattern recognition receptors. In particular, recent research has focused on TLRs, toll-like receptors, which play an important role in the recognition of pathogens and other substances. Alcat detects and measures changes in these phagocytic cells, mostly neutrophils, in order to qualify and quantify an innate immune reaction to a variety of foods and chemicals. Neutrophils have a very short lifespan and are extremely aggressive after they are activated. They can destroy pathogens in many ways, primarily by bombarding them with cytotoxic oxygen and nitrogen radicals through the production of acids such as hypochlorous acid or through the activation of proteolytic enzymes including myeloperoxidases. As an inflammatory cascade arises, the immune system stimulates granulocyte activity through the activation of pattern recognition receptors located in the cell membrane and in the cytoplasm of immune system cells. This activity becomes part of a vicious cycle in which chronic activation of the innate immune system results in persistent inflammation and oxidative stress factors that produce and perpetuate disease symptoms. Sustained activation of these immune cells is destructive, promoting effects ranging from microinflammation of intestinal tissue to the type of chronic widespread inflammation responsible for numerous disorders, including gastrointestinal complaints, metabolic disorders, skin diseases, obesity, respiratory diseases, neurological, and musculoskeletal disorders. Researchers have shed light on a unique mechanism of cell lysis that's triggered when neutrophils attack, the ejection of neutrophil extracellular traps, commonly called nets. Nets are composed of a mixture of reactive cell contents and neutrophil DNA. Nets can rapidly disarm and immobilize pathogens until they can be removed by macrophages. Thus, pathogenic bacteria are literally captured in the net, where they are immobilized and rendered harmless. The DNA and nets from activated neutrophils must be cleared by the body so that they don't perpetuate an immune response. The inflammasome is a multi-protein complex that, when activated, sets off a cascade of events related to chronic inflammation. This protein complex becomes activated in the presence of PAMPs and DAMPs. The inflammasome promotes maturation of pro-inflammatory cytokines, so basically it is found at ground zero of the immune inflammatory response. The inflammasome is directly related to chronic disease, especially those related to food and metabolism, including chronic bowel disease and metabolic syndrome. Researchers recommend the Alcat test as a screening tool for identifying foods that may activate the inflammasome and trigger this chronic inflammatory response and related symptoms. The inflammasome influences a number of processes in the body that can ultimately disrupt metabolism. Functions and systems affected include antimicrobial defense, the immune response of the mucosal membranes, especially in the gut, which can critically alter composition of intestinal microflora. Expression and activation of metabolic syndrome, which is essentially the comorbid occurrence of obesity, metabolic dysregulation, insulin-resistant, and chronic inflammation. The inflammasome affects insulin signaling in the islet of the pancreas through inflammation-associated deconstruction of insulin-producing beta cells. These negative effects ultimately promote the transition from obesity to type 2 diabetes. Here you see the progression of the inflammatory cascade that is orchestrated by the innate immune system when presented with the endogenous or exogenous triggers. Diverse stimuli, including endogenous factors such as food particles, stress, damps, and exogenous factors such as PAMPs can trigger reactions of innate immunity. Reactions are conducted via signaling pathways and activation of specific pattern recognition receptors, PRRs, ultimately resulting in inflammation. The Alcat test recognizes blood cell changes, especially those occurring in polymorphonuclear PMN cells. Sustained inflammatory reactions weaken the entire organism and produce or exacerbate myriad symptoms or disorders. To detect and characterize food sensitivities, the Alcat test describes the reaction of cells to food in terms of changes in cell volume, size, and number. It basically detects, quantifies, and qualifies granulocyte activation. The Alcat test detects all three types of leukocyte reactions towards food ingredients and chemicals. These distinct reactions are automatically measured by the RoboCat2 and transferred into a reaction profile qualifying reactions as severe, moderate, mild, or non-reactive using a specific algorithm. In 2015, Yale School of Medicine was first to demonstrate that both DNA, acting as DAMPs, and myeloperoxidase of peripheral blood leukocytes were released during certain food reactions, and that these items were identified with the Alcat test. The clinical conclusion drawn from the research is that the Alcat test can demonstrate a clear correlation between A, an Alcat reaction, B, a specific test item, and C, the pathogenic inflammatory effect. Sensitivity to food was originally referred to as a toxic idiopathy by founder of immunology J. Friedman in the early 1920s. We now know that different arms of the immune system will generate different types of reactions. For this reason, an individual may have an adverse reaction to a substance, but not have an antibody-mediated specific allergic reaction to it. Adverse reactions not mediated by the specific immune system or antibody formation are defined under the terms intolerance or sensitivity. Even these terms have distinctions between them, as you will see. Remember, allergy refers to an antibody-mediated response of the specific immune system. The term allergy is not used to describe sensitivity reactions mediated by the non-specific innate immune system. The Alcat test addresses mechanisms of the innate immune system and does not measure antibodies at all. As you can see here, characteristics and measurable immune response differs between food allergy and food sensitivity. IgG antibodies reflect an immune parameter of the specific adaptive immune system. However, there are no conclusive studies indicating that antigen-specific IgG antibodies truly indicate an adverse reaction to food. Of the 4 subsets of IgG antibodies, IgG4 has been discussed most often with regard to representing an adverse reaction to food. However, researchers currently conclude that the presence of IgG antibodies is more reflective of tolerance to certain food or substances rather than intolerance. In fact, IgG antibodies are believed to block specific IgE antibodies, reducing severity of an IgE-mediated response. Both IgE and IgG4 are dependent on a Th2 cytokine profile. Thus, all antigens that induce an IgE response are, in principle, also efficient IgG4 inducers. In contrast to IgE, food allergen-specific IgG4 is not an indicator of a classic food allergy or a food sensitivity, but a natural physiological response of the immune system to food allergen exposure. Therefore, increased IgG4 levels are the expression of a chronic exposure to allergens and a marker of tolerance induction. Research shows that elevated IgG4 levels in childhood are later associated with a tolerance towards food, not with an intolerance. This means that elevated IgG4 levels do not indicate a pro-inflammatory response. The term adverse reaction to food is a general premise referring to all excessive body reactions to food, ingredient, or additives. Such reactions are subdivided into classic allergic, non-classic allergic. The latter category has been referred to simply as food intolerance until recently. Within the food intolerance category, a pathophysiological distinction is made between the following types: genetic, such as celiac disease; pathophysiological, an enzymatic or lactase deficiency perhaps; pharmacological, may be induced by bioactive agents; immunological would be adverse reactions induced by the immune system; psychosomatic symptoms or diseases may be considered an intolerance. In 2015, Professor Alessio Fasano proposed going farther and suggested a more precise approach by assigning non-classic allergic reactions to two different entities, food intolerance and food sensitivities, and clearly defining them. According to the United States National Institute of Allergy and Infectious Disease, food intolerance occurs when the body lacks a particular enzyme to digest nutrients. Nutrients are too abundant to be completely digested, or a particular nutrient cannot be properly digested. Therefore, symptoms are exclusively GI and mostly secondary to sugar fermentation by the intestinal microbiota, leading to production of gas that causes abdominal distension, abdominal pain, and irregular bowel movements. This table will help you understand the differences between allergy, intolerance, and sensitivity. As you can see, the mechanism behind the adverse reaction varies between classic wheat allergy, specific immunity, celiac disease, also specific immunity, and gluten sensitivity, non-specific immunity. A classic wheat allergy is mediated by IgE antibodies and considered a food allergy. Reactions to gluten itself, however, were found to be mediated differently from classic allergy. A 2010 study determined that celiac disease and non-celiac gluten sensitivity were separate entities with different mechanisms of pathogenesis. Therefore, different underlying causes for adverse reactions to gluten. Non-celiac gluten sensitivity was proposed to result from non-specific innate immune response to gluten-containing foods, whereas celiac disease was associated with a specific adaptive immune response. Hence the differentiation between an allergy, intolerance, and sensitivity to a food can be illustrated with the example of adverse reaction to wheat. The Alcat testing method is controlled at many levels. It is performed with strict preparation protocols and quality assurance guidelines in an FDA-registered medical device establishment using an ISO EN 13485 certified medical device manufacturer. Blood samples are carefully processed, starting with expedited courier service to ensure the samples arrive while blood is still viable. Effector cells, such as neutrophils, have a lifespan of about 5 days. Accordingly, leukocytes are analyzed within 30 to 72 hours of blood collection. Pre-analysis includes standardized methods are used to separate leukocytes from red blood cells. The leukocyte population is mainly composed of neutrophils, eosinophils, basophils, macrophages, monocytes, and lymphocytes, though present in different quantities. A preliminary test is first performed to determine cell number and distribution. The patient's white blood cells and plasma mixture are introduced to individual test substances. For test substances and cell confrontation, test substances are carefully prepared with strict protocols and quality assurance guidelines. The manufacturing division is an FDA-registered medical device establishment and is also ISO EN 13485 certified as a medical device manufacturer. Cell Science Systems Lab is certified and licensed by the United States government. Quality controls are in place. All operations from instrumentation, health, to quality control testing, to data integrity verification are constantly monitored in real time and displayed on screens throughout the laboratory. These systems are HIPAA compliant and provide traceability, validation that all of our processes and procedures are performed according to protocol. Rest assured that Alcat Test is strictly controlled and implemented via exceedingly high standards. Measurement of leukocytes can now take place. Here you can see all the various components of blood. Alcat testing detects all white blood cells, leukocytes, and records their distribution based on size. Reactivity is measured by analyzing changes in their number and volume. The reaction of the leukocytes as immune effector cells of the innate immune response is detected primarily by changes in leukocyte cell volume and or cell number. Alcat measures changes in cell volume, form, and number. Cell volume increased, usually associated with when an immune stimulus promotes an effector cell response that involves formation of mediators. Cell volume decrease is usually reflective of degranulation, onset of apoptosis, and autophagy. Cell number decrease is indicative of apoptosis, netosis, necrosis, or pyroptosis. Remember, the Alcat test analyzes the effect of each individual test substance on the leukocytes. Cellular immune reactions lead to changes in cell volume and cell number, which are displayed in histograms after mathematically computerized algorithm evaluation. Thus, the degree of reaction is exactly calculated and mirrored as a snapshot in 4 reaction degrees: severe, moderate, mild, no reaction. Reactions are displayed on a single-page result sheet using a simple color scheme, red, orange, yellow, green, so that the patient receives a structured and easy-to-understand overview. Almost 500 individual substances can be analyzed with the Alcat test. Flexible test profiles combine food, chemical, additives, medicinal herbs. All are available. Let's look a bit more closely at the measurement technology that Alcat uses. The measuring principle of the RoboCat2 device is based on the fact that during the passage of a cell through an electric field, the resistance as a function of the cells will change in proportion to the cell's volume, impedance. The system's processor will capture the change and express it as a pulse, where its amplitude is directly proportional to the cell's volume. The number and size distribution of the cells is displayed graphically. For relative analysis, the degree of deviation of the measurement to the control curve, baseline, negative control, is determined mathematically using computer algorithms. It is then categorized into 4 response levels: severe, moderate, mild, or negative. In this graphic, you see that the Alcat RoboCat2 analysis correlated closely with other accepted technologies, namely CytoViva and FACS Caliber, another flow cytometry method on the far right. Remember, the Alcat test is a leukocyte activation test that measures cellular immune responses following exposure to individual food substances at the molecular level. Cellular changes are associated with pro-inflammatory reactions, triggered primarily by degranulation, processes of granulocytes, the main fraction of leukocyte population. Degranulation processes are an early immune response to danger signals. Granulocyte reactions are thus a clear indicator for the sensitivity to a substance. Alcat is able to detect these cellular changes via means of a measurement methodology known as impedance cytometry. Alcat measures response to greater than 450 individual items, including foods, medicinal herbs, pharmaceutical agents, additives, colorings, and molds. The resulting Alcat report helps individuals eliminate the foods and substances most likely causing an innate immune response, hence reducing immune reaction and inflammation associated with these triggers. Alcat's measurements technique is a specific combination a flow cytometry and impedance methodology. According to the current state of science, it is used to analyze early cellular responses and is superior to all other available methods for the analysis of cellular response. Analysis by conventional flow cytometers can miss early adverse cellular response. We refer to the work of Professor M. Cooper, a pioneer in label-free technology for continuous, non-invasive, real-time cell monitoring. The Alcat test is analytically and clinically validated. Analytical validation results from both clinical studies and from approval, certificates, and patents. Cell Science System Corporation maintains CLIA certification and is registered with the FDA, further demonstrating analytical validation. Cell Science System can also provide proof of compliance with FDA current GMP standards. Clinical validation of the Alcat test for the identification of sensitivity to food, ingredients, additives, and other components is demonstrated in the number of studies. The abstracts of more than 40 studies on the Alcat test are documented in the appendix of the comprehensive dossier, Scientific Basis of the Alcat Test. The overall study material provides a reliable overview of the Alcat test as a useful tool in clinical practice for guiding the treatment of patients with food sensitivity and inflammation-related diseases. Understanding sensitivity and specificity of the Alcat test will further demonstrate clinical validation, as you will see in the next slide. The clinical validation of an in vitro laboratory method is mainly characterized in two ways: A, the diagnostic sensitivity, and B, the diagnostic specificity. The diagnostic sensitivity is the percentage by which the test correctly identified those with the relevant sensitivity to the tested food or additive when compared to all individuals. In short, the people with an identified sensitivity are actually recognized as a true positive. Diagnostic specificity reflects the proportion of the detective true negative test subjects, those who have no sensitivity to the tested compound. The assessed clinical sensitivity and specificity of the Alcat was determined to be 83.4% for foods and 96% for colorings, which is remarkably high. There was a high correlation between positive reactions and severity of disease. For example, healthy asymptomatic subjects were less reactive versus those with symptoms such as hay fever, allergic rhinitis, asthma, irritable bowel syndrome. Those tested more reactive. The meta-analysis of approximately 1,300 patients showed that modifying the diet according to Alcat test results led to positive improvements in many common symptoms and chronic diseases. The Alcat scientific dossier provides over 40 abstracts addressing the science and validity of the Alcat test. Alcat test results are easy to understand using a clear structure, making review easy for both patient and practitioner. In order to ensure patient compliance, follow-up is important and includes detailed personal instructions and optional support of nutritional counselors for implementing dietary changes. Food sensitivity test report based on an extensive list of foods along with special sections regarding gluten, includes gluten and gliadin component as well as individual grains. Milk products includes casein and whey components as well as different milks. Sugar intake includes candida albicans and different sugars. Chemical sensitivity report includes food additives, coloring, flavoring, sugar alternatives, preservatives, environmental chemicals, pharmaceutical substances, molds, and bioactive agents. Medicinal herbs functional food report basic, medicinal herbs, functional foods, adaptogens. Performance profile, athletes, supplements. Women's profile, menopause. Personalized food to avoid list. Personalized rotation concept. Guidance notes include directory of test items. For reactive foods, a 3 to 6 month rotation and elimination period is recommended. The goal of the Alcat protocol is a reset of immune reactivity. Unlike allergies, the innate immune system, which does not involve antibody formation, operates without memory function. The absence of exposure to the substance may result in resolution of the sensitivity. Thus, reactive foods can often be tolerated again after the defined elimination period, basically promoting a reset of immune reactivity. We obviously need to eat food to live. However, much of what we call food today is vastly different than what humans had consumed for thousands of years. While foods are principally derived from vegetable and/or animal sources, foods today contain a number of substances, including man-made chemicals, that may be intentionally added or may result from food processing. A standard fast food meal may contain more than 100 artificial substances, including colorings, preservatives, environmental chemicals, artificial sugars, and pesticides. Cosmetics and the environment can also be major sources of chemical exposure. Cleaning products, cigarette smoke, pesticides, and vehicle exhaust are significant sources. So even people who eat primarily organic foods are not exempt from exposure to additives and contaminants. This array of preservative, hormones, drug contamination, antibiotic, xenobiotics, flavors, flavor enhancers, spices, colorings, stabilizers, the list goes on, are suspected of causing considerable health problems, and researchers suggest that approximately 20% of symptomatic patients may be chemical sensitive. These chemical substances can lead to malfunction of biochemical pathways in the body, disruption of the intestinal flora, and both localized and systemic inflammation. These disturbances can result in a variety of symptoms, both physical and psychological symptoms. In fact, symptoms are typically multisystemic and can affect cognitive function as well as musculoskeletal, gastrointestinal, and cardiovascular systems. As you can see, many chemicals and food additives may be hidden and quite difficult to detect. These include the list you see here. Here you see a sample of the scientific research that has been conducted on the Alcat test. A comprehensive overview of the scientific research conducted to date on the Alcat test can be found in the detailed scientific dossier. Research confirms the reproducibility, sensitivity, and specificity of Alcat as a clinical tool for identifying foods and compounds that may trigger immune cell activation and inflammation. Studies include double-blind, placebo-controlled, molecular pathomechanism assessment, mechanistic technical studies, and review papers. Conditions studied include gastrointestinal disorders, skin diseases, respiratory problems, metabolic diseases, autism, migraine, and others. Double-blind placebo-controlled food challenge is the gold standard for diagnostic testing. In the double-blind placebo-controlled food challenge, certain foods are eliminated from the diet and individually reintroduced at predetermined time intervals. Because symptoms are often delayed, an objective assessment is the only reliable way to identify a true connection between food consumption and symptoms. Neither the physician nor the subject knows whether the food used in oral provocation were identified by the Alcat test as positive, reactive, or negative, non-reactive. The double-blind, placebo-controlled food challenge design was used to determine that results of the Alcat test were comparable to the results of oral provocation. These results are described in greater detail in Alcat, a new cellular test for food sensitivity presented at the annual meeting of the American In Vitro Allergy and Immunology Society, August 1990, Toronto, Canada. Here you can see that even early on in the history of the Alcat test, physicians, researchers achieved positive results when correlating Alcat test results to patient symptomology and presentation. Detail and citations of this research, along with additional international double-blind study oral challenge research, scientific review papers, and case study reports can be found in the Alcat scientific dossier. Yale research conducted from 2014 through 2017 confirmed that Alcat test was able to identify foods that were associated with measure innate immune activity and inflammation. Details of the published studies can be found in the comprehensive scientific dossier. Cell Science Systems puts 30 years of research, development, and experience with the Alcat test to use. The company holds a number of patents and currently operates worldwide. The company laboratory and equipment are under stringent regulation and consistently meet established guidelines and requirements, as you can see here, including CLIA certification. Cell Science System has researched and validated the principle that an ongoing immune response of the innate immune system to foods and its ingredients leads to an uncontrolled immune activation causing chronic inflammation. The relationship between an overactivated innate immune response and various chronic inflammatory diseases is now recognized, making the Alcat test indispensable in the clinical setting. Thank you for joining me in this webinar, and I hope you now see the value in using Alcat testing in your practice for positive patient outcomes.